Hello. Hi everyone at the Addictive Brain. This is Maurice at BenchSci. I am now, so first of all, this is my first time ever doing a live session, so welcome any feedback. So I'm now sitting in the hallway of our office, and just want to show you guys, starting by showing you guys what our office looks like. We're located in Toronto, Canada. Today is extremely cold. I just survived probably the worst winter this year coming to the office. But okay, let me start by switching the video view so you can see our office. Okay, there we go. So this is the front entrance of our office. You can see we have a nice little logo right on the door. So again, we're based in Toronto. We're a startup from University of Toronto. So let's just walk through the office. Oh shit, I think I locked myself in. Okay, no, there we go. Okay. So you can see this is our front foyer. We have 3 meeting rooms right now. This place used to be just like a reception area, but we grew so much we needed another space for a meeting room. And you can see that each of our room we have a name. So for those of you science geeks out there, this room right here is called the lysosome. And our biggest meeting room, our boardroom right here, we also have a pretty suitable name for it as well. We call it a nucleus, which is the most important thing really in a cell. And so it's this meeting room to us. And you can see we have a smaller meeting room right here, which is where I will come back later to show you guys our technology. We call this room the Goji. And you can also see this is our office space. You can see this is pretty good outside. There's not too much cloud, but it's extremely cold by the way. Yeah, and so this is where our space is. Currently holds about 40 people. We started out with 5. Now we are about 40. We expect to expand to maybe closer to 60, 70 by the end of this quarter. You can also see there's a little space right there at the corner. That used to be our entertainment room. We had a punching bag, we had some PS and a TV sitting in there. But again, we grew so much, we needed another space for more people, so we took that away. But hopefully that'll come back as we renovate our office next, I think, in quarter 2 this year. And right at the far end, we have our kitchen area, and that's where we name it the mitochondria, where we get our energy from. So I thought that's pretty suitable as well. Yeah, so I think thanks for you guys who just joined right now, by the way. So that's our office. It's pretty cozy, but again, we hope we can expand it so we can accommodate more people in the upcoming year. So switching back to me, hi, hey guys. So I'm gonna just step into a room right here, grab a seat. Yeah, so I understand that for some of the audience, some are not scientists. So I'd like to just quickly explain what we're doing here at BenchSci. So I guess first, let me start by just giving a quick intro about myself. My name is Maurice. I finished my PhD in pharmacology from the University of Toronto in 2015, so about almost 4 years now. And so I did a lot of basic science experiments as part of my PhD, and what I needed oftentimes is a reagent called antibodies. So to give you guys a quick concept about what experiment is like, so I think a typical analogy would be, so for the scientists doing experiments in the lab, it's very much like when you guys cook in the kitchen. You put a whole bunch of ingredients together and in a certain proportion, and then you end up with the results. So that's very much what things are like when we do experiments in the lab. And a lot of times when you do experiments or when you cook, you need those ingredients, and you need a way to decide which ingredients you should use. So for example, if I were to cook some Chinese and I want to get some soy sauce, but there are so many soy sauce out there in the market, and how do you know which ones will work? Because they can all be added to your food, but they may not taste as good. So you need a way for you to be able to determine, okay, which soy sauce I should get. And so I guess oftentimes what you do is you go online or you hear from friends, like for recommendations, for reviews, and that review process, that comparison process, is exactly what the scientists do as well when they need reagents for their experiments, so the things that they need for the experiments. And the typical place where scientists get their recommendations or reviews on reagents is they go to scientific papers. So a lot of times when people found certain interesting findings, they write papers and they publish it in different journals, and their data are then out there in the public for people to review. And so that's the place where scientists typically go whenever they need reagents. They go to papers to see, okay, which lab has used certain reagents, what was the data, does it make sense for me to use that in my experiment? But this is where sort of the problem comes up, because there are so many papers out there. I think it's been estimated there's about 28 million papers since 1980s, and this number has been growing, like it's doubling every 9 years. So because there's so much information out there, it's very difficult for the scientists to be able to find what they need from this massive amount of information. And that's also, I guess, one of the problems that I have experienced myself as I was doing my PhD. So that is why when I first met the co-founding team at BenchSci, which is mostly also scientists at the bench, we started discussing this idea about how we can make this information more accessible, how we can make sure whatever data is out there in the literature can be easily found by the scientists whenever they need to look for reagents for the experiment. And that's really the idea about BenchSci, is digesting all this information out there, make them more, make a sense out of it, and then deliver and then present the most relevant data to the scientists as they need it. And the way we do this is through machine learning technology. Essentially, we teach a computer to read papers the way that we would as a scientist, and then we simply feed the computer with a lot of information, all those papers as they come out, and then it will simply extract the relevant information that we wanted to extract, and then we have them available on a platform, which is a free platform actually for academic scientists. So we support basic research from academic institutions, because in the end that's where we came from. And I guess I would like to also talk a little bit about antibodies. Of the reagents that we're working with, we're starting out with something called antibodies. And antibodies is one of the most commonly used reagents in basic research. And why is antibody important? So I thought of another analogy. I'm not sure if it's the best one, but imagine you're reading a book. Like, you imagine reading a book that has like 300 pages, and then you had to stop midway. And you typically what we do is we put a bookmark at page 250, and make sure that next time when we come back, we can still start from page 250. You want it to be at page 250. And what antibodies is doing is similar thing. It's like there's a whole bunch of proteins in a particular sample, and then use antibodies to bookmark a particular protein that you want to study. And the expectation is that if you're using antibody for protein X, you want to be able to detect and see protein X. Same thing as if you bookmark it at page 250, you want to be able to come back at page 250. But the problem with the antibody is that it's not always specific. Sometimes it might detect the wrong target. This is because antibody is made typically from an organism, so it has a biological basis. So there's some degree of variability. So the difficult thing about antibodies is that sometimes you use this in your experiment, you expect to detect protein X, but it may not. As if you put your bookmark page 250, and next time you come back it's at 260. And that's extremely annoying, and it doesn't really. All of a sudden, things don't make sense at all. So that's the problem that scientists are facing as they use antibodies, because sometimes that would happen. And the best way, at least in our opinion, the best way to overcome this is to be able to make sure that you find out how specific antibodies are being used by different labs. You do this from looking at papers, and then review those information to see, okay, should I use this antibody for my experiment? And again, that's what we're doing at BenchSci. We use our machine learning technology to decode the literature to find out how antibodies are being used. And then we make that information available on a free platform that people can essentially come here and search for the target, and then find the information that they need to decide which antibodies they should purchase. So that's I would say a quick overview. And I'm not sure if anyone here has any question. If not, I think I would like to move on to showing you guys a quick demo, like for now. I guess this is a part for the scientists out there, to show you guys what we have built and how you can leverage this technology for your experiments. So let me switch the screen again. Oh, there we go. Okay. Yeah, so you can see, by the way, we here at BenchSci, we drink coffee using a beaker. So you know exactly how much caffeine you're consuming every day. So I thought that's pretty cool. So I'm not sure how you can see this, but so yeah, I'm just showing this screen of my laptop right now. So this is the website that we have, and this is freely accessible for any scientists out there. If you work in universities, hospitals, government agencies, nonprofit, you can simply access this platform for free online. You don't have to install anything. Everything is on the cloud. So the first thing we ask people to do is ask them to create an account, because we do have information that's from closed access publishers, and so we cannot show everything to the public without first knowing that they're from a research institution. So, but regardless, it's a free process. So you simply come here using your university email, and you'll be able to sign up for free. Straighten the screen like this. Okay, so once you have created an account, you can log in with that information. And for me, I just log in with my utrono.ca email right here. And yeah, so once you log in, you can see this screen right here. Actually, I have this. I'm not sure if this is better, but actually, I'm gonna try this here. So just like very much like a search engine screen, where you see a search bar at the top, and this is where you can start by inputting whatever target you're interested in studying. Say if I am interested in studying the protein PCNA, I simply come in and then I type up the protein name, and here we have a whole bunch of options to help you decide which specific PCNA from which animal origin you're interested in studying, and you can go ahead and select any of the options here. So what you will notice here is that for each of the names, we have actually also different aliases that are associated with each of the proteins. And this is because a lot of times, I think scientists were really bad in naming proteins. So the same protein can sometimes be referred to by so many different names. So in order to avoid any ambiguity as you search for your target, we have spent quite a amount of time to go through different databases, linking together different aliases back to the gene level to make sure that it doesn't matter how we refer to the protein, you'll be able to see the right target. And say let's just go with PCNA in general. And once I select that, so as I mentioned in the beginning, what we're doing at BenchSci is that we use a machine learning algorithm to screen the literature to find out how antibodies are being used and present the data here on the platform. And so you can see right after you search for your target, you can see all these different figures right here from different papers, along with the title of the paper, year, and author, and so on, where we present all the data that's relevant related to PCNA right here. And of all these different figures, you can see actually there's quite a large amount of data for PCNA because it's quite commonly studied. There are close to 5,000 figures related to PCNA. So the immediate question you may have is, okay, how can I make sure I find the information that I want? There's no way I can go through the entire 5,000 figures. So that's the reason why we have all these filters on the side. So you can see here we have figure usage data. Sorry, let me just put this down. For example, we have filters for contexts that are described in the papers, including the different techniques, organism, tissue, cell type, and so on. And then we also have filters for the antibody specifications, such as the basic information about the host, reactivity, clonality, and so on. And the idea here is to be able to help you find the information that you need simply by applying the filters that match your experimental context. So a lot of times when I look for antibodies, I need to make sure that it has been used in the same technique that I plan to use, because depends on how your sample is treated, the antibody may or may not detect the right target. So this is typically where I go first. I go ahead and take a look at, okay, so there are all these different techniques right here, from immunostaining, Western blot, and so on. I go ahead and select a technique that I plan to study, say immunofluorescence. And once I do that, I'll be able to filter through the figures gradually. So here, first thing you see that now the numbers went down from 5,000 to close to 900. And you can see from the images here, they all have immunofluorescence staining in them as well. And so what I can do further is to be able to apply additional filters to further narrow this down from 900 to a more manageable number. So say if I'm interested in studying a particular tissue, I go ahead and apply a filter here as well. So say lung. Then by simply applying two filters, immunofluorescence technique and a lung tissue, I narrow the figures down to just 39. And of all these different figures right here, I can also choose to sort them by either the publication date or impact factor. So I can filter by published date to see what is the most recent. You can see we have data from 2018 oncoimmunology here. Or I can sort them by impact factor to see data from the top journal first. So we have here Nature Medicine right here at the top. And for each of these figures, you can click into them to further review the data directly on the platform. So you can see the immunofluorescence experiments right here, along with the figure legend at the bottom, so you get a sense of what's going on in this experiment. We also have a link that can take you to papers from here to the publication, to the original publication. So if you are interested in studying take a look at the full method or other data, you can go back to the paper right there. And for the figures, you can also choose to share with your colleagues or bookmark them and save them into different folders so you can organize them and review them later on. And the most important information that we want to pull out, which is the antibody that was used to generate this figure right here, is just shown right beside the figure. So you can see we have this antibody used in this paper is from DACO. So if I click into this antibody, I'll be able to see even more information about this particular product. So first of all, we have the name at the top, and then we also have the product information right here. So you can see the SKU is M0879, the target is PCNA, the clone is PC10. And you can also see that at the bottom from this chart here. It also gives you an overview about how this antibody was used across different papers. So you can see this antibody has been used 119 times, immunostaining, IHC 71, IF 45, Western blot 30, and so on. So this gives you a sense of, okay, how are other scientists using this antibody, and should I use this for my own experiment? And if you go further down, you'll be able to see more figures here. So these are figures that were generated from by the same antibody, and currently we're looking at 210 that were generated. And this is where you can also apply the filters to narrow this down. Say if you're interested in also seeing the data from Western blot, then I can also filter that right here. And then I'll be able to review the data again on a platform. And say after reviewing all this information, you decided to purchase this antibody, then there are two options that you can do from this point on. So for some of the antibodies, you can go back to the vendor website to purchase them. So this is taking you back to the original manufacturer website, PCNA antibody. Or for some of the antibodies, if we have partnerships with a vendor, then you can also purchase them through BenchSci, through our platform directly. And this is where you can simply check out using shopping cart right here, and then complete your purchase. So yeah, so that's very quickly on high level what we do here at BenchSci. Somebody asked if we can elaborate what we do. So I briefly talk about that in the beginning, but I'm happy to say that to say that again. That means it switches back to the screen. Yeah. So maybe I'll sit on the other side. So what we do is, as you just saw, we have a platform that's designed to help scientists look for antibodies from the literature. And the way we do it is we have developed a machine learning technology to be able to read the papers and then find out how antibody was used, which antibody was used, what was the data, and then we present the figures, the published data, on the platform for you to review that information. And then if you found a data that makes sense, that had pretty good data, then you can find out which antibody was used there, and go ahead and purchase that for your experiment. So that's what we do at BenchSci. So I'm not sure if anybody has any question about either what we do, about antibodies, about myself. Happy to answer them at this point. Any question? No? Okay. So in that case, if you have any other question that come up afterwards, you can always reach out to me by email. So my name is Maurice. You can find me at Maurice, Maurice, at BenchSci.com, and happy to take any questions there as well. So thank you all very much today for joining me to this session, and thanks again to the Addictive Brain to give us the opportunity to be able to present here to everyone here. And yeah, thank you all very much. Take care now.